So, good morning, everyone. Thank you for attending today's webinar on magnetofaction for neurosciences. I am Sophia Murray, Sales and Marketing Project Manager at OSI Biosciences, and I will be your host for today. So, I will leave you with the presentation and see you at the end for the Q&A session, hosted by Cédric Sapé, a biology researcher at OSI Biosciences and a specialist in this product line. Do not hesitate to ask questions along the presentation, and we will do our best to answer as much as possible at the end.
OSI Biosciences is a French biotech company founded 20 years ago, specialized in molecular delivery systems present in France and the US. The French site is located in Marseille and comprises the headquarters and the production site, divided in two laboratories over 500 square meters. OCBSAS is responsible for R&D and production, mRNA and LNP platform, marketing, and sales. The U.S. site is installed in San Diego and deals with marketing, sales, and business development. Over the years, we have settled a worldwide distributors network. One of our objectives is to develop the spreading of our reagents and expertise. Regarding transfection of neuronal cells, several methods can be considered, but even if they each present some interest, they are not optimal for neurons transfection. Electroporation or nucleofection are very efficient transfection methods with a rapid and high transgene expression, but they induce high toxicity and can be applied only on freshly isolated neurons still in suspension. Moreover, cost is really expensive. Viral infection is maybe the most efficient way to genetically modify cells. However, it is technically challenging for the creation of the viral vectors, for example, impose hazardous handling and can be very expensive. On the contrary, calcium phosphate, one of the oldest way to transfect cells, can be used. It is applied to adherent culture and present a low toxicity when optimized. However, the efficiency can be very low with a moderate reproducibility. Finally, lipofection and polyfection are often used because they are simple and quick transfection procedures that require low optimization. They show some reproducibility, but they present high yield of toxicity with a low to moderate efficiency. The problem is that neurons are sensitive to cytotoxicity and difficult to transfect with most methods. This comes from the fact that postmitotic neurons are sensitive to various parameters, such as microenvironmental or pH changes, osmotic shock, chemical, or also physical shocks. As we can see, any activation or shock can lead to the presence of granulations on apoptotic dendrites. Moreover, when dealing with the neural stem cells, transfection methods can interfere with proliferation and differentiation. This challenge has inspired us to develop effective transfection methods based on magnetofection to transfect neurons and neuronal cells. It was used by Borelli et al. In their original paper in Nature Protocol in 2007. The paper would set up the basis of genetic modification of neuronal cells using magnetofection. This method would become one of the most efficient and non-toxic physical gene delivery systems. Magnetofection is a simple and highly effective method to transfect primary cells and hard-to-transfect cells. Its principle is to associate nucleic acids, transfection reagents, or viruses with specific magnetic nanoparticles. The resulting molecular complexes are then concentrated and transported into cells supported by an appropriate magnetic field. In this way, the exploitation of a magnetic force exerted upon gene vectors allows a very rapid concentration of the entire applied vector dose on cells, so that 100% of the cells get in contact with a significant vector dose and promotes cellular uptake. The The principle of magnetofection is also a really simple and rapid procedure, yet very effective. First, nucleic acid solutions are mixed with magnetic nanoparticles in a serum-free condition. The complexes are formed after incubation, 20 to 30 minutes at room temperature. They are then added to the culture medium of cells, and the plate is positioned onto a magnetic plate, delivering a specific magnetic field for 20 minutes at room temperature or inside an incubator. Finally, the magnetic plate is removed and the cells are incubated under standard culture condition until evaluation of the experiment, generally from 12 to 72 hours. Then magnetofection allows long-lasting expression up to 96 hours or a week and sometimes more. Now let's take a look to see what happens after the complexes are added to the cell culture medium. When the cells are placed onto a magnetic plate, the specific magnetic field represented here by the B vector attracts the magnetic nanoparticles onto the cell surface within minutes. Under the influence of this physical force, the magnetic nanobeads are clustered and enter the cell through classic endocytic pathways, such as Clathrin or Cavelin pathways. Importantly, no hole is created, leaving the cell membrane intact as opposed to other physical methods such as electroporation. Inside the cells, the complexes are then located in the endosome from where they will evade. The nucleic acids will be transported inside the cytoplasm and/or the nucleus, while the beads will be excreted out or be degraded by the cell's iron metabolism. This unique technique allows to bypass the classic transfection limits, as most of the dose comes into contact with the cell surface within minutes. The endocytosis of the complexes is triggered by the clusterization of the beads on the cell membrane. Magnetofection limits the diffusion and allows to overcome the pore adhesion occurring depending on cell type. This method is really efficient and adapted to primary and hard-to-transfect cells, such as neurons, as illustrated here. Considering neurosciences applications, we have developed mainly two transfection reagents for nucleic acids, Neuromag and Gliomag. Neuromag is dedicated to any kind of neuronal cell culture, from hippocampal or cortical neurons to astrocytes, whereas Gliomag is more specific to microglia cells. The magnetoception technology has several advantages compared to existing transfection methods. It shows a high transfection efficiency with any nucleic acid. Indeed, it allows to increase the efficiency from 30 to 500%. It is a very powerful tool for hard-to-transfect and primary cells. High performance can be achieved even with low dose of nucleic acids. The concentration of genetic material onto cells induce an acceleration of kinetics, and the beads are biodegradable iron oxide nanoparticles, which renders this method safe and universal. Over the years, Neuromag has built its reputation on three pillars. It is non-toxic, versatile, and most of all, highly efficient. If we take a look at the toxicity generated during transfection, it appears that it increases with the dose of nucleic acid for a given ratio and with the ratio of delivery vehicle for a given nucleic acid amount. Actually, to obtain the maximum efficiency, using a classic transfection reagent, high dose of DNA is needed that can induce high toxicity. When using magnetofection, as the nucleic acid dose needed to obtain the same efficiency is reduced, the toxicity threshold is not reached and thus the viability is increased. In other words, as magnetofection is more efficient, less material is used and the cell viability and integrity is preserved. As we begin to consider this earlier, Neuromag is a versatile transfection reagent that can address any kind of brain cell, from cortical neurons, hippocampal neurons, or neural stem cells with any kind of nucleic acids. We can show you some examples on primary rat cortical neurons at DIV7, primary mouse hippocampal neurons at DIV7, but also at DIV20, on neural stem cells, human hypothalamic cells, and N2A mouse neuroblastoma. The polyvalence of neuromag is first observed when transfecting hippocampal and cortical neurons at various days in vitro, or DIV. Multiple publications have used neuromag to transfect neurons from DIV0 to DIV22. For example, we can cite Asselin et al. That transfected cortical neurons at DIV0 or Charrier et al. At DIV15. As mentioned earlier, neuromag efficiency and biodegradability enables to vary the ratio of reagent used and to lower nucleic acid amount favoring viability. This is especially true for sensitive neurons at early or late DIV. Neuromag versatility means we can address any type of neurons. For example, primary motor neurons at DIV2 were co-transfected using neuromag to follow axon outgrowth and study signaling pathways in amyotrophic lateral sclerosis. Another model, amygdala neurons, were transfected with luciferase reporter, and as we can see here, luminescence in transfected amygdala neurons was as high as in hippocampal neurons, and this allowed the authors to identify novel polymorphic region that control BP4 activity. Remarkably, neuromag is also efficient for neural stem cell transfection. In 2011, we have developed a specific protocol to transfect neural stem cells, as we can see here. The efficiency increases with DNA amount, from photo A to D. The fact that neuromag did not induce toxicity and did not impair cell differentiation was emphasized in 2020 by Finch et al. That demonstrated a high viability rate of 92% and a long-lasting expression up to 4 weeks in transfected NSC. Moreover, the authors showed that daughter cell types of engineered NSC neurons, astrocytes, and oligodendrocytes were also transfected. Neuromag efficiency and reliability is demonstrated by studies of Derek Welsby et al., who start in 2013 by using neuromag to screen an arrayed library of 1869 cRNA against 623 kinases to cover the mouse genome in retinal ganglion cells. Derek was not left alone during this preliminary study. Our technical department supported him and his team the best they could to set up their protocol. The authors chose neuromag because, as they stated, traditional transcription procedures were toxic or non-effective with retinal neurons. Only neuromag was thus able to transfect their cell model, as we can see here. Later in 2017, the same authors performed genome editing experiment by transfecting a pool of cRNA and cgRNA, and 1 year later they applied the same protocol to fresh retina isolated from postnatal mice. All their experiments with neuromag were concluded in 2020 by a patent describing a method for treating retinal degeneration. This kind of story is, of course, what we wish for all of our customers. An interesting feature of magnetofection, beside classic gene expression, silencing, or genome addition, is the possibility to co-transfect with good efficiency primary neurons. And depending on the way the complexes are formed, the outcome of the experiment is different. If the nucleic acids are pooled and transfected neurons receive the same material and co-transfection occurs, all the neurons that are transfected express both DNA. When formed separately, complexes transfect one or the other neuron, leading to independent transfection that are very useful to study excitatory pathways, for example. Finally, more than versatile and non-toxic, neuromag also outperforms its main competitors. For example, for primary hippocampal neurons, we see that efficiency lasts from 24 to 96 hours when others fail to transfect or lose their efficiency. Neuromag outperforms competitors also on cell lines such as B65 or C6. Consequently, the number of publications Mentioning the use of magnetofection for neurosciences application is still growing since 2005 when comparing on 5-year periods, and 500 publications will be achieved soon. If we take a look specifically to the publications mentioning neuromag, then we can see that most of them relate to primary cells. The publications highlight its versatility since articles cover the majority of the brain territories and other territories as well. As a matter of fact, top 5 neuron cells are cortical, hippocampal, motor neurons, neural stem cells, and retinal root ganglion neurons. The most majority of the cells transfected comes from 3 species: mouse, rat, and human, but other species can be addressed as well as xenopus, chicken, bird, monkey. And if any cell from any territory from any species can be transfected, neuromag can also deliver any kind of nucleic acid from DNA, representing most of the publications, to mRNA and cRNA, or even large bacterial artificial chromosomes. Another important population of cells that regulate brain development, maintenance of neuronal network, and injury repair are the microglia cells. They serve as brain macrophage and were considered hard to transfect cells. As neuromag, gliomag has built its reputation on 3 pillars. It is non-toxic, able to knock out genes, and most of all, highly efficient. Opposed to classic transfection methods that provide low efficiency and induce cell death and/or activation of the microglia, gliomag has established itself as the reagent of choice for transfection. As stated by Carrillo Jimenez al. In 2018, gliomag is an easy and effective method to transfect primary microglia cells with cRNA, inducing neither toxicity nor inflammatory activation. The toxicity graph shows no impact of magnetofection on these cells' death, and no dramatic inflammation following magnetofection. As we can see on the graphs, more than 93% of microglial cells are transfected with cRNA when using gliomag, leading to a massive gene silencing, whatever the target. Gliomag was demonstrated by Smulders al. To be very efficient in cell lines such as BV2 cells. Magnetofection is superior compared to other chemical transfection methods such as lipofectamine 2000, extreme gene, and calcium phosphate, as we see on the pictures in the graph. Gliomag outperforms the other transfection reagents for BV2 transfection. As for neuromag, the number of publications reporting the use of gliomag to transfect a variety of microglial cells is rising every month. The magnetofection technology presents a unique feature associating physical force and chemical compounds that gives it specific advantages. By playing with ratio of magnetic nanoparticles, for example, it is possible to address various models. The same reagent can be used for different cell types. Once formed, the complexes are very stable and protect their cargo. The magnetic field attracts a whole dose within minutes, rendering the method compatible with any cell culture medium, model, or system. Unique to magnetofection, the transfection medium can be replaced right after or hours after transfection. Leaving the cell plate onto the magnetic field during washing procedure induces the complexes to stay onto the cell surface. All the unbound material, excessive nucleic acid for example, is removed, increasing the viability. As this transfection method is non-toxic, since the material is biodegradable and low dose of nucleic acid is used, thanks to a specific protocol, long-term cultures can be transfected. And finally, the magnetofection system is compatible with serum-containing medium. There is thus no need to perform a medium change before, or culture the cells in absence of serum. The only requirement for magnetofection is the transfection reagent based on magnetic nanoparticles and a magnetic plate specifically designed for this application. The magnetic plate is a one-time buy and completely reusable, so you do not need expensive equipment, contrary to approaches such as electroporation or gene gun. 3 magnetic plates are available. a supermagnetic plate that can be used for any applications, a magnetic plate with 96 individual magnets for 96 wells cell culture, and a megamagnetic plate that can accommodate up to 4 culture dishes at the same time. The plate can be washed with ethanol 70% and used within incubators or robots. Neuromag and Gliomag are part of a complete range of reagents for a wide range of solutions, from bioproduction to viral application, protein delivery, mRNA or LNP production, and of course, transfection. All the reagents developed and sold are under the ISO 9001 certification. These innovative reagents allow OZ Biosciences to offer solutions in 3 main areas: the cell and gene therapy, bioproduction, and custom platform productions. All the processes developed at OZ Biosciences are assisted by a supportive technical team of experts that you can reach by email at tech@ozbiosciences.com for Europe and Asia, or techusa@ozbiosciences.com for the American continent. What I would like to emphasize before ending this webinar is the fact that since the first paper in 2007 describing the magnetofection method to transfect primary neurons, hundreds of articles were published in high-ranked journals, Neuromag being mainly used. This success is due to the fact that magnetofection, and to a larger extent Neuromag, is non-toxic, reproducible, allows long-lasting expression of transgene, is very versatile, allowing to transfect any nucleic acid in any cell from any origin and work from DIV0 to DIV22.
It's the end of the webinar. Thank you very much, Joanna, for this presentation. And thank you, everyone, for your attention. You can visit our website by scanning the QR code or contact us by email at contact@ozbiosciences.com for Europe and Asia, and contact USA at ozbioscience.com for the American continent. So we are now going to go through the questions that were sent during the presentation. So the first one is what happens to the magnetic bits once they are endocytosed by the cells?
Yes, so first it is important to mention that each component of our magnetic bits is totally biodegradable. So either the magnetic bits will be degraded or digested, or they will remain in kind of vacuole inside the cell until they are secreted. And in our lab, we estimated that the bits remain 4 days to a week inside cell before the beginning of degradation that occurs through the iron pathway. And also one point that I would like to add is that the bits that remain in the cells are totally compatible with following experiments such as PCR or Western blot. And I think this is quite important.
Okay, thank you. Second question is, what are the main differences with classical transfection method and related toxicity?
So magnetofection has many advantages over other transfection method, and one of them is that it uses less nucleic acid for an enhanced efficiency, because it concentrates the nucleic acid onto the cell surface. And so when low doses of nucleic acid are used, there are mainly 2 consequences. The first one is that we need less transfection reagent to perform transfection, and also is that the cells are getting less activated, and thus they do not go apoptosis because the threshold, the toxicity threshold, is not reached. Another feature we are repeatedly seeing is that with magnetofection, we can remove medium right after the transfection procedure, because magnetic field keeps the particle onto the cells, and we can remove all the inbound material, and that also allows to reduce a lot toxicity.
Okay. Another question was, is magnetofection applicable to in vivo experiments?
Yeah, it can totally be applied in vivo. It is usually used to transfect focal zone, precise zone in the animals, thanks to specific magnets. And we offer various specific magnets from large one to very small one. And if we consider neural application that use magnetoaffection in vivo, it has been demonstrated for viral transduction with lentiviruses, adenoviruses, and also for gene silencing, sorry. And specifically, when talking about Neuromag, it was used to deliver mRNA and DNA in rat and mouse brain, and because its completion capacity are very high, it allows to reduce the injection volume.
Okay. Another question was how to choose the best ratio plasmid quantity, such as shown in tips and troubleshooting slides.
There is no rule on how to choose the best ratio and the most efficient DNA dose for a specific cell type. In most cases, Neuromag and GlialMag standard protocol works very fine, but sometimes the efficiency is not optimal and needs to be optimized. And this depends on many parameters, such as, of course, the cell type, the neuron type, or the DIV, and also the culture condition, and of course, the plasmid, because depending on the plasmid, its size, the way it was purified, its promoter, the efficiency may be different. And so what we generally recommend when performing magnetoaffection is to optimize the condition for a specific cell type by trying various amount of DNA with various ratio of transfection region. And what I generally recommend is to have a look at our citation database to see if an experiment was done with the neuron type and the specific protocol to do it.
Okay. Another question, what's the actual composition of your Neuromag and GlialMag products?
They are made of, let's say, two major components, an iron oxide magnetic core, which confers the super paramagnetic property of the magnetic bits, and also a cationic layer that surrounds it and allow to stabilize the particle. And this layer will complexes the nucleic acid and allow to release them once the particle is inside the cell. In the case of Neuromag, the layer is based on proprietary lipid, and in case of GlialMag, it is more polymer.
Okay. We are going to take a last question. Don't worry, all the questions that were asked are going to be answered by email at the end of this webinar, so we will get back to every one of you. So the last question will be, what are the size of the nanoparticles?
As nanoparticle diameter are of GlialMag and Neuromag are between a range between 150 and 180 nanometer.
Okay, thank you very much, Cedric.
Thank you, Sophia.
So this is the end of our webinar. As I said, we will get back to every question that weren't answered in this Q&A session. And don't hesitate to contact us if you have any questions. Thank you very much.